Welcome back to biology. We are still working on Unit 3C, where we're looking at cell division. Today we're going to focus on DNA replication. The big unit idea stresses that the structure of DNA stores genetic information for cell division, differentiation, and maintaining in general of all organisms. But in order to be able to do that, to make new cells and cell division, we have to be able to replicate or duplicate the DNA that we have accurately. So the two learning targets that we have for this lesson is to determine the accurate pairing of nitrogenous bases. In other words, nitrogen bases like adenine and guanine and cytosine and thymine. I feel like you guys have that down from what we covered in class as an intro. We're going to review that briefly. And then the second one is to explain that nucleic acids store information about how to build and run an organism. And I think we're going to be spending a lot of our time on that because that is basically the first part of the central dogma of heredity. So let's get started. Just an update, I have been keeping a running list of the vocabulary terms that we've covered so far. I am not saying that these are going to be something you have to know the definition of for the test, but when I put a term on this list, it's important that you understand how these terms all work together. I know it's almost like a new language, so if you type these up into a Word document and you type them or you upload that Word document into one of your Quizlet websites, oftentimes they'll generate the definition for you. You can tweak it if you want to, but it's a good place to start. Also, there's going to be some GIFs that are interactive that you may want to consider adding to your team's Padlet. The first thing to do in order to hit the main target is to focus on determining the accurate pairing of nitrogenous bases. And again, I feel pretty confident that you're able to do this. We've discussed it in class. You showed me that you could do it. So all I'm going to do at this point is summarize what you guys completed in class as a group. The first thing we're going to focus on is to circle a nucleotide. We're going to label the sugar and the phosphates that you would find in the DNA backbone. And then we're going to make sure you can pair up those nitrogen bases, A, T, C, G, or adenine, thymine, cytosine, and guanine, on the 8 bases. I'll be happy if we can just put the first 4 in. And then after that, we're going to fill in this complementary sequence, which would be the end result of replication of DNA, is to make a whole sequence. So the first one was circle a nucleotide. Okay, so remember that a nucleotide is the house with the flag and a balloon. So again, the house with the flag and the balloon. So during class, one of the things we corrected was many of you wanted to just select one of these structures. And I understand that because you want to participate, but you may not know the right answer. If you need to review nucleotides, go back to Unit 2 for biomolecules and look up nucleic acids, because the house with the flag and the balloon is the structure that we use to recognize nucleic acids, or in other words, nucleotide. The next thing that the directions ask us to do is to label the sugar and the phosphate. So I thought the easiest way for us to do that on this presentation would be to just draw a line from the sugar. Now remember, sugar is a carbohydrate. So carbohydrates, when we draw them, again, from Unit 2, should be geometric shapes. And in this case, it's a 5-carbon sugar. So you're looking for the pentagon. It's a 5-sided polygon. That's what we use to represent the sugars in carbohydrates. And then the other structure to look for was phosphate. I can find my pen again. All right, so the phosphate were all of these little black circles. And we're using circles because just like with ATP, if you remember that the phosphates are a phosphorus and 4 oxygens. So when you draw a circle around that chemical structure, it ends up, you can make a circle from it. That's all. It's just easy for us to remember. And we like the analogy, the house, the flag, and the balloon. And the balloon needs to be round. Okay, then the next part was to identify and label the base pairs that match the existing ones you see here. So adenine always binds with thymine, and thymine likewise binds with adenine. Guanine is going to bind with cytosine, and cytosine with guanine. And I'm not going to finish the rest of them, but you can see that anytime you see A for adenine, you need to think T for thymine in DNA. It'll change for RNA, but we'll get there later. All right, and the last thing was to write a complementary sequence. So for when you see A down here in this long strand, it's basically what we just did. It was just practicing your ability to do that for a long strand. So once we filled that in, that was the sequence. So that sequence of nucleotides, remember, will code for specific proteins. All right, moving along. This series of questions, after the lesson that we're talking about right now, you should be able to thoroughly answer these prompts. So I planned on working through these together as a group. We're going to be answering these throughout. We've already answered some of them in prior lessons. So we're going to tie it all together. When you come into class on Monday, I want you to be prepared to participate. And I'm going to ask you all these questions, and I want you to chime in with groups to figure out what the answers are. Alrighty? So let's get started making sure we are comfortable with them. The big idea about DNA and how it holds information that's responsible for cell division and maintaining cells, etc. It all ties into this idea called the central dogma of genetics, or the flow of information. So there are several steps. Today's focus is going to be on replication. All right, it's going to be talking about replication of the DNA itself. Before you can divide as a cell, you have to make sure you have enough of everything to have an even number. So if you remember from the cell cycle, the S phase of the interphase portion of cell division is where S for synthesis, another word for synthesis is replication or copying, is what we're focusing on, how that happens. So I am showing you this so that you can see that the reason we can say that DNA holds all the information is because after it's replicated, when the DNA gets transcribed into a copy of that information called RNA, and then translated into the language of proteins, that protein is what's going to be the substance that either exhibits a trait that makes you blonde-headed or blue-eyed or have lactose intolerance or any of those beautiful imperfections that make you you. So this is the grand scheme, the central dogma, the flow of information of genetics. DNA replicates itself. It's transcribed. And when you think about transcription, think original to copy. So a DNA to an RNA. So I want you to have in mind a job like a medical transcriptionist, those people who take what the doctor says in gobbledygook doctor gibberish speak and transcribes it into something that you and I can read on our papers that we take away from the doctor. And those papers, the equivalent of those would be RNA. The RNA tells us what to do. So the RNA is what a ribosome will use to translate this information into another language, another chemical language, from nucleic acids to amino acids to make a protein. So I want you to think of a translator who literally takes those instructions and translates them into another language for somebody to understand better. Before we can do any of this, you need to understand how this first part happens. So that's what we're looking at today. Replication just means making a copy of itself. And I just want to remind you that replication is happening here in S phase. This is where replication happens. So make a note of this on your notes, that replication is happening in the S for synthesis phase. The end results would be two exact copies that the cell will split between its two new daughter cells. Why do we do that? Because all cells need DNA. It's the blueprint of life. They need two copies before they can divide. If without it, you don't have any directions. So there are some steps, specific steps, but they can be simple if we don't look too hard. DNA, remember, is a double helix. It's a twisted ladder for a more layman's term. So when you look at the structure of the DNA, there are two strands, and those two strands are twisted upon themselves. We have to untwist them in order to access these nucleic acids in the middle. You have to be able to open this zipper to expose the teeth that are the flags or the nitrogen bases in order to match new nitrogen bases to them. If you brought nitrogen bases in here to try and match them, these are happy already. They're already bonded. They don't need to form a new attraction. So until this is opened up, you won't be able to make any new DNA. So first you have to unzip. There's a special player, an enzyme, that will do that. I'll introduce that to you in a minute, but just keeping in mind, you unzip the DNA or unwind it or unravel it, whatever word you prefer. And then each of those two sides of the zipper are going to be a template. And you know, a template is what you use to make a copy that stays the same. The new nitrogenous bases, the new bases, A, T, C, or G, are going to be brought in and properly matched to the existing template strands. So the new ones are going to be called complementary base pairs. And then since both of these strands, the original purple in this case strands, exist once you've gotten replication completed, they call this semi-conservative. You are half keeping the old strand, the original strand. So keeping that in mind, that's a term I'm going to review with you. So what prevents many mistakes when copying the DNA? There's a lot of ways that we can combine different bases, and we know that amino acids have a specific sequence to make a protein, a specific protein. So we have to get from a specific sequence of DNA to a specific sequence of RNA to a specific sequence of amino acids to make the protein. Well, there's a lot of possibility for errors in that way. So think about what prevents most of the errors that we would see from happening. Well, I've added in some notes here for you. One of the ways, the most important way that we can prevent mistakes when copying the DNA is these base pairing rules that you just learned. Oopsie. So these base pairing rules that you just learned, as long as the DNA gets copied following these rules, everything should stay in the correct sequence. Sometimes mistakes still happen. Complementary is when two things go well together or pair well. So for example, if you're into accessories, boots and belts. If you're not, peanut butter and jelly. So you always want your purse to match your shoes and your belt, right, ladies? Or if you're going to eat something like a sandwich, you want like peanut butter and jelly or hot dogs and mustard, whatever, okay? DNA replication in detail. So what do you need to know for the test? You are for sure going to need to know some of these more important players, like DNA helicase. So helicase is the one that unzips the helix. Look at this root word here, helicase, helix, right? So it takes the helix, the DNA double helix, and it unzips it, okay? So it unzips this. That's why it's a helicase. ASE endings are enzymes. So in this case, DNA polymerase. It says protein, but in this case, it's a special protein. It's an enzyme. Polymerase. Look at that word. Polymer, you've learned in the second unit. Polymer means a large chemical structure, and it's a chemical structure made of DNA. So it builds a DNA molecule by bringing in new N-base pairs to add to the existing ones and build this structure. DNA polymerase pairs nitrogen bases with the old strand. So what you need to understand is that the two new copies of DNA, so you can see those down here, right? Here's one, and here's the other. These are semi-conservative. Semi-conservative. And that's because this original strand is still included with one old. So one original and one new. And that's partly conserved DNA. This is a little GIF that I like that shows you the process of the unzipping. So I don't know if I can write on this or not. Oh, I can. But there's your helicase. Okay. Helicase is going to unzip. This structure here that forms is called a replication fork. Replication is the process. The fork is where things divide, right? The two polymerases you can see are building the new DNA. So this red new DNA here is the new strand. So those are the two enzymes that you need to focus on. Here's the illustration I was trying to show you in class today. What this includes for you is the terms leading and lagging strand. And where's my header? Okay, so here's your helicase again, going this way. It's going to make a replication fork. And then this little guy here and this little guy, I can't keep them. Those are the ones building your DNA strand. It doesn't matter that you know that it's a 3 to 5 or 5 to 3, although if it helps you to understand all that, you can review that in the part of the lecture from the video that's going to be tacked on. All right, here's the diagram showing that you're going to have to be able to summarize this. So step 1 needs to include helicase. Okay, so helicase unzips or unwinds, depends on how you look at it. In step 2, the new nitrogen bases are coming in. And then in step 3, so this would be DNA polymerase doing that. And in step 3, 2 new semi-conservative DNA strands are formed. Okay, so that in a nutshell is the very least you need to be. Oh, and you know what you need to remember for this, right, is the base pairing rules. So let's add that on here. Where's my, okay, just got to find my marker here. That follows the base pairing rules. Sorry it's messy, but you can do your own. All right, what characteristic of DNA replication, so what component, function, process that we talked about is the most important in how the original genetic code is preserved from generation to generation. How does DNA prevent making mistakes? Ooh, so there was one thing we talked about, the primary safeguard or safety feature are the base pairing rules. And if you don't remember what those are, let's write them down again. A, adenine to thymine, or vice versa. So let's make this a double arrow because it could be starting with thymine. And guanine to cytosine. What are those words she keeps saying? These are the nitrogen base pairs that we learned about at the beginning. There are 5. The one we're not bringing in here yet is uracil. You'll learn about that in RNA. All right, and so that's all the new stuff. So you need to be able to participate in class on Monday when we talk about these things here. Where do you find a gene? You find a gene on a chromosome. It's a segment of DNA. I am really roughly writing this down for you, but you're going to take your own notes. What does a gene hold instructions to make? So it's instructions for proteins, right? So, and proteins have so many functions in your body. That's 2 words, believe it or not. So many functions, okay? What's the relationship between a gene and a trait? A gene is the information for a trait. What's a trait? Example of a trait would be like a physical characteristic of you, right? So could be an intolerance. It could be hair color. Could be height. Could be lactose intolerance, etc. Can you identify where replication takes place? Now, what you need to remember is replication takes place where, but when it happens in the S phase, which is interphase, and interphase is happening in the nucleus before mitosis occurs. The 3 steps, in order to summarize that, because I don't have a lot of space, I would ask you to go back to this, right? So when you summarize these 3, make sure you know the helicase enzyme, the DNA polymerase enzyme, and then the base pairing rules. Make sure you mention all those. Can you name 2 enzymes that play a main role? So I just did that for you, right? I did helicase and polymerase. There will be an RNA polymerase, so maybe you just be fine if you memorize polymerase. So basically know what these very gist of these prompts are about. There is an animation, and I will link it when I link this video into your content down here. It'll be in SafeTube, so you won't have to deal with what you call it. You won't have to deal with YouTube, but yeah, these are the terms that the animation is going to refer to. The ones in red are the ones you're going to want to know. So know the ones in red. All right. So yeah, make sure that you're comfortable with these success criteria. If you can do these, then these ones are going to be a snap for you, because this is the more specific examples for how to hit those learning targets. And that's all I have for you. I don't have any structured notes for you. I do want you to take notes. You're going to want to pause the video back here where I talk about this step, but in your own terms and cleaner. This question, these questions, which are basically the end ones. And then again, feel free to snip anything you want from the pictures. I hope that this has helped when you're prepping for replication. Have a good weekend. I'll talk to you next lesson.